Yes, thank you, Enzo, for the great introduction. So hello, everyone. I'm Veronica, and I'm part of the FAMI group. So together with Eike, I will be sharing this first session today. And so for the first speaker of the session, I'm happy to introduce Gagan Gupta from the Indian Institute of Technology, who will be talking to us about fairness in skin image classification, as well as clinical text representations. You have the floor.
Thank you. Uh, so let me just bring up my slides. So this is a work that we have presented at MICAI this year, and it's about fairness for the skin disease classification. So dermatology has a skin color problem because the data sets that are available, there is a lack of darker skin even in the textbooks and journals, and that leads to lack of understanding of how the skin diseases look on the darker skin. And even for AI or the ML models, they're not performing very well when it comes to the darker skins. So, so here's a brief outline of what we did. We leverage graph optimal transport as a means to align the image representations with the clinical textual representations of the disease or the skin conditions. And this we found that reduced the bias and the disparities in the skin disease diagnosis, which I'll share some results on. And we have analyzed Fitzpatrick 17K, which is a broad data set, internet data set, but also has noisy images and even mislabels. And DDI is a much cleaner and clinically validated data set. We have considered three fairness metrics, PQD, DPM, UM, which I'll be describing as I go along. So this is how the Fitzpatrick scale looks for skin. We have on the one hand the white skin, which always burns and never tans. And then at the extreme, we have the black skin, which is heavily pigmented, never burns and tans very easily. So there's a whole range. And then there are conditions which are named the same way, or they're the same thing, like acne or sunburn or some kind of melanoma, which is a cancer, but its appearance on these different skin tones will be different. So when I look at the data distribution, I can see that the darker tones have less samples as compared to the fairer tones. Both in the Fitzpatrick, that is what we see. And there are three broad categories: benign, malignant, and non-neoplastic. Whereas in DDI, I have malignant and non-malignant. The problem with DDI is it's a very small data set, only 656 images are there. So it's a more clean and clinically validated data set. Fitzpatrick has more images, but it is noisy and there are mislabels. So just a pictorial data distribution. There are some tones for which I have more images. There are the darker tones, T5, T6, I have less number of images. And this is just some visuals from how the images are looking. So the question here is, given the skin image, the AI/ML model has to predict which type of skin condition broadly they have, whether it's benign or malignant or non-neoplastic. So more details are there. In detail, we had 114 different skin conditions in the Fitzpatrick, which we would leverage, and then we will group them into these three broad categories: malignant, non-neoplastic, and benign. The DDI, as I said, is a smaller data set. So we are grouping the skin types. T1, T2 is one group for us, T3, T4 is another, and T5, T6 is another. Again, some examples of how the DDI images look. So our problem formulation is as follows. If we are given a RGB skin image, which has a height, width, and three channels, we want to model the probability of Y, which is the skin condition, given the image X, without being affected by S. And in our case, the sensitive attribute is the skin tone, the T1 to T6 on Fitzpatrick that I was showing. So this is the overall architecture. we are given an input skin image. We are training a image encoder. The image encoder is generating the patch embeddings, and we are using these patch embeddings and aligning them with the text representations of the detailed 114 skin conditions which are in the Fitzpatrick. And we are using a text encoder. We are using a large language model here to generate the text embedding. And what happens is that these text embeddings themselves have some relationships. So various types of cancers which are malignant, the text representations of those, if you visualize in this TSNE plot, we see that they are closely related to each other in the embeddings. So these embeddings are closer to each other. Their dot product is higher. Whereas the other conditions, like the benign conditions, they have their own cluster, and non-neoplastic is a bigger spread. So the whole idea is that in the text representations, the relationships between the various skin conditions are present, and how can we leverage them for learning these better image embeddings. So that is what we are doing. The text encoder is frozen, the embeddings are generated once for all, and then they are guiding us in terms of how to learn the image embeddings or the patch embeddings. So there are three branches in the architecture. The main contribution that we have done is in the alignment branch, where we are aligning the patch embeddings to the text embeddings. And I'll focus on that. The target branch, uh, the branch one, is just a standard cross entropy loss based classifier where we just predict the skin condition. The sensitive attribute branch is interesting. It's based on another paper that we have sort of extended. Here, the idea is to suppress the sensitive attribute, which is the skin tone, and there is a interesting gradient reversal layer which suppresses those features which allow a classifier to distinguish between the skin types, because we want to learn the skin, how to predict the skin conditions, not the skin tones themselves. And here is the new thing, which is the graph optimal transport. And basically, it deals with these distances. So on the left side, I have, let's say, the images, which is one domain. And here I have text, which is another domain. And think of some examples like images or patches and the words or the disease names that are present in both groups. So there are these types of inter-class distances, like these two, x and x prime are two images between which I am looking for a cost, how close or how similar they are to each other. Here I'm looking for the cost between two diseases, how close are these two diseases. And then there is this distance, the Gromov-Wasserstein's distance, which is on these pairs. So x prime, y prime, they are the corresponding, like x prime corresponds to y prime, x corresponds to y, and therefore now we are wanting to preserve the relationships between these two texts and their corresponding images. So there's Wasserstein systems, which uh, provides a mapping between the image and the text, and the Gromov-Wasserstein distance tries to preserve the relationships among images and the relationships amongst the corresponding text. So there is an intra-graph cross matrix CX that we compute. Similarly, CY we compute for the other domains. So X could be image, for example, Y could be text. And then there is a cross-graph cost matrix, which is the Gromov-Wasserstein, sorry, the Wasserstein. And both are sharing the same transport plan. So the transport plan can be thought of as this, that suppose you have a distribution or some quantity that you want to, let's say this is some commodity that I want to transport from these locations which are in green to these, which are the production nodes, to these consumption nodes where there is a requirement or a demand for this. So I want to ship this quantity that I have, or this commodity that I have, to these consumption nodes in a way that my transportation cost is minimized. So here we want to align things. When we are aligning images to text, we want to align them in the way that my dissimilarity, which will lead to this cost functions, these CAB, C1 x prime, C2 y prime. So I want to align them in a way that my cost functions, when I multiply the distribution or the weight in the transport plan to this cost, the overall cost of whole transport is minimized, which means that similar things should align to similar things in both domains. So this is what we did. But there is one more interesting thing that we did was to learn a mask over them. So because if you notice these images, when we are looking at these, natural images, the disease may be present in only one of the patch. But what about all other patches? Where will we align them to? So they are like noise. And therefore, we want to learn a mask which tells us which patches are important so that we want to align them. And the alignment loss should only be computed on this mask. But this mask is also learnable, so there is no annotation of the image in our methodology. The mask is also something we are learning on the fly as we are learning how to generate the patch embedding. Similarly, we are also learning how to generate the mask. In other words, those patches which have an interesting embedding, which look like diseases, we would pass them to the alignment branch. But those patches whose embedding is like noisy and they don't really look like diseases, we would try to suppress. So this is the intuition, but there is a math behind that, where we have put it in our loss functions. I'll not go over the math in so much detail right now, but just the intuition is that we want to mask out those regions which are not very interesting. And let's now look at the fairness metrics. So one of the metrics we work out is this predictive quality disparity, where we are looking at the minimum accuracy achieved over a skin tone was divided by the maximum that we achieve on a skin tone. So in this example from DDI, I have grouped them into three groups, T1, 2, T3, 4, and T5, 6. And Fair Disco is a previous state-of-the-art paper which achieves these accuracy of 88.6, 71.7, and 92. So the minimum was 71.7 among these blue bars, and the maximum was 92. So the PQD was 78. Whereas for patch align, the algorithm that I just presented, not only are we improving the accuracy, so each of these purple bar is bigger than the blue bar, it's taller. So we have improved not only the accuracy, but also the fairness because the min over max ratio has now grown to 87%. So that is the basic idea that we see, that with the help of this alignment, we are able to improve both the accuracy as well as the fairness because the group which was performing the best is now doing better as compared to the best one. Similarly, there is another interesting metric, which is equality of opportunity, where we are not looking at only the broad, accuracy, but also the true positive rate across groups. So we have malignant, benign, and non-neoplastic as three different types of skin conditions. And now we are computing this min over max true positive rate for each of them. And then for our technique, this comes out to around 79%. And in a later table, I'll show you how it compares to the other algorithms. But we were able to see that EOM also improves, not only just the accuracy. And there's also DPM, which I'll not go over. So let's study this results on the in-domain classification of Fitzpatrick, where we are, learning or training our algorithm on a mix of all the tones and then evaluating also on all the tones. So patch align our algorithm achieves the highest average accuracy, but also quite high in terms of this equality of opportunity metric. There is also a multitask learning that we had implemented as a comparison, and that also does well. I didn't describe it here, but my focus was more on patch align. And then this also is an interesting result, which is out of domain classification, where we train on two tones. For example, in this row A, I'm training my model on T1 and T2, but evaluating on the other four tones. And here from Fair Disco, on average, we improved like 4.5 or 5% improvement on the average accuracy. And in the PQD and EOM metric, not on the EM, but on the PQD metric here, there is improvement. And for the remaining all of the experiments B and C, there is improvement on all the three metrics. So fairness is a bit tricky thing. We cannot always get high fairness in all experiments, and there's also high variance that we noticed. But on accuracy, the results are more stable. And interestingly, even if we don't give any examples of T1 to T4, let's say in this third experiment, we can still perform fairly well.
So,
So
sorry to
this is
interrupt,
sort of
but
the last
could you
slide.
could, oh, right.
This is the last slide.
Perfect. Perfect. I
Yeah.
was looking at 27 out of 35, so I was just wanted to give you a heads up.
No, those are some just backup slides. So yeah, this I
Okay,
end my time talk here. It's
perfect. Very nice. Thank you so much for the talk. I don't know if we can we're clapping virtually or with sound. So yeah, so now we have a few minutes for questions. So if you have questions for the speakers in the Zoom, underneath there's a section Q&A. So this is not the same section as the chat. And then in the Q&A, we can see the questions show up, and then I and later the other chairs will um, tell the questions to the speakers. So while the questions are coming up, I would like to start. So I'm really interested in these data sets. I've worked myself a little bit with Isaac and with Pad Ufesh data sets. So it was I was quite interested that you said that this DDI that it was
yes,
a smaller size, like a few
yes,
magnitudes smaller.
yes,
So, but that it was like more accurately labeled. So, what is your
yes,
impression like on the general trade-off of size and quality? Do you think we need larger and larger data sets to achieve good results, or
yes,
is a small
yes,
but
yes, we do. Yeah, I mean, like, so this is like interesting, right? Like, so either you make very big, and then we can tolerate the noise, but also see in DDI, it was not divided into three skin conditions. We only did malignant and non-malignant. So it would be better if we had, higher quality data sets, at least, of the order of few thousands. So that is always better.
but
But we didn't have,
given the choices we
yes.
have, what would you choose?
Like a larger and noisy versus a smaller and a clean one?
Yes, and maybe what would the proportion difference be, let's say?
It's a very tough question to answer because we don't know the test set where it would come from. So usually when we are doing these data sets, both our train and test is from the same data set. But
Hmm.
if the test comes from a totally different one, then it's harder. So I would just like guess that it would be like a bigger and a broader data set would be better, just so that we can cover more at the test time.
Okay,
So
thank you. There is a question now from one of the participants from Theo Sauget from ITU. And the question is, do you think that adding skin types to the skin labels would also help mitigate the biases instead of penalizing the correct prediction of skin type to remove the information from the learned representations? For example, having a label melanoma of a T6 skin type and melanoma of a T2 skin type. So as I understand, those would be then distinct
it could,
labels.
yes, that could, because what we know from the literature is that the appearances of the diseases on different skin types are different. So, actually in these data sets, even the skin tone may not be correctly given. So, although in the ground truth I have been given T4, it might be T5 according to someone else. So, there's always some noise on the tones themselves. But there is something like that, that the appearance of the diseases are different on the different skin tones. So, it
Hmm.
could help to provide this explicit information while training.
Yeah, and a related question, I guess a clarification from Mary Lucas. Are the skin type categories determined visually?
Yeah, visually. So it is like human annotated. And there are also some automatic techniques to give to predict the skin tones. There are
Okay.
limitations to both of them. I mean, nothing is perfect.
All right. Thanks a lot. So in the interest of time, let's thank the speaker again.